Huperzine A has been reported to have several neuroprotective effects. In addition to the potential brain-protecting effects, preliminary research also shows that Huperzine A has some effects on specific cognitive functions like learning and memory. For example, an early study showed that Huperzine A appeared to enhance memory and learning in junior middle school students who are complaining of memory problems. The Huperzine A has been reported to be relatively well tolerated and showed promising treatment potential in patients with Alzheimer's disease and vascular dementia, according to an early study. Parkinson's and Alzheimer's disease patients experience degeneration in the nucleus basalis, a region of the brain that's extremely rich in acetylcholine cells. According to one study in an animal model, the Huperzine A was reported to help to restore the healthy brain electrical activity. In animals who are suffering from lesions in this brain region, the Huperzine A also reduced the impairments in cognitive and memory functions caused by brain lesions. Huperzine A possibly increases acetylcholine, specifically by inhibiting acetylcholinesterase, the enzyme that's responsible for breaking down and removing this neurotransmitter from the brain. It potentially increases the level of important growth factors like nerve growth factor, brain-derived neurotrophic factor, and TGF-beta, and these compounds are believed to help to protect the brain and might even possibly improve memory and learning ability. Although Huperzine A's full mechanism of action is not yet completely known, early results from one animal study in mice where it reduced blood flow in the brain, or chronic cerebral hyperfusion, suggest the Huperzine A treatment can alleviate the cognitive impairment by reducing myelin damage, decreasing inflammation via TNF-alpha, alleviating white matter lesions, reducing the overall cell death and damage, and preventing and reducing the negative effects of low oxygen or hypoxia. Limited clinical trials suggest that Huperzine A helps with learning and memory, possibly by increasing the neurotransmitter acetylcholine. Citicoline is an important intermediate in the creation of phosphatidylcholine from choline. In some studies, the citicoline has been reported to improve memory and learning, including situations of impoverished environments, and improve cognitive frontal lobe function in humans. It was shown to reduce memory impairments associated with brain diseases in humans, including chronic cerebrovascular disease, dementia, cerebral insufficiency, memory problems, dementia, and ischemic stroke. Some of the potential mechanisms that have been suggested to be behind the effects of citicoline include enhancing membrane phosphatide synthesis, decreasing pre-programmed cell death of neurons or apoptosis, and increases of levels of antioxidant compounds like glutathione, while decreasing the levels of oxidized glutathione. Panax ginseng is a traditional Chinese herb, and it's been claimed by some to improve long-term memory. At least one early human study has reported that Ginkgo biloba and ginseng in combination improves memory in healthy adults. The Ginkgo biloba has also been reported to have positive effects on dementia and Alzheimer's disease, as well as long-term memory in general. Although scientists don't know exactly how Panax ginseng works yet, preliminary findings suggest it can improve memory by reducing oxidative stress by promoting the release of nitric oxide, which protects the brain, by increasing serotonin, and increasing the level of various important neurotransmitters like dopamine, epinephrine, and norepinephrine that are involved in learning. In addition to the general mechanisms, some other potential mechanisms that have been suggested to be behind the effects of Ginkgo biloba include increasing acetylcholine, which can theoretically help with learning, attention, and memory, and increasing antioxidant enzymes like superoxide dismutase and catalase, as was seen in a rat's hippocampus. Protecting the mitochondrial membrane and preventing cytochrome C release, preventing neurons from pre-programmed cell death by the process called apoptosis, and protecting against amyloid beta-induced toxicity, which is believed to be one of the important factors in the development of Alzheimer's disease. According to a handful of early research studies in both animals and humans, supplements containing Bacopa Monnieri have been reported to improve attention, cognitive processing, and working memory by inhibiting acetylcholinesterase, improving the overall memory and general cognitive function based on the auditory and Stroop tasks, improving free memory recall, improving delayed word recall, improving memory acquisition and retention in older men, and improving memory formation and retention. Some of the potential mechanisms that have been suggested to be behind the effects of Bacopa include increasing acetylcholine levels by inhibiting acetylcholinesterase and or choline acetyltransferase, and glutamate levels, each of which is a neurotransmitter that's believed to play a key role in learning and memory, and by increasing enzymes and transporters that act to increase serotonin levels, which can also play a role in overall learning and memory. By protecting the brain from oxidative stress by increasing the levels of antioxidant enzymes like superoxide dismutase, catalase, GPX and GSR, as well as glutathione, one of the brain's most potent natural antioxidant compounds. By increasing the brain blood flow by stimulating nitric oxide production and by inhibiting calcium fluctuations. These mechanisms are believed to be vital to memory and to overall brain health. According to some research, some early evidence suggests sodium butyrate can place the brain into a state that could potentially benefit long-term memory. For example, sodium butyrate was reported to increase memory function in patients with age-related brain disease, according to an early clinical trial. Another study has reported that exercise and sodium butyrate together might be able to help weak, newly formed memories get transferred into a more stable long-term memory. Similarly, another study reports sodium butyrate can help prevent memory impairments in rats who were subjected to experimental pneumococcal meningitis. According to one animal study, the sodium butyrate has been reported to boost long-term memory by increasing synaptic activity in the hippocampus, a region of the brain that's believed to be critical for forming and storing new memories during learning. Some of the other potential mechanisms that have been suggested to be behind the effects of butyrate include increasing brain-derived neurotrophic factor, nerve growth factor, and GDNF, and stimulating the growth and the development of nerve tissues. Early clinical trials showed that butyrate might enhance the long-term memory. Creatine's main role is recycling cellular energy in the form of ATP, or adenosine triphosphate, primarily in the brain and the muscles. Creatine is currently in phase 3 trials for the treatment of Huntington's disease. Creatine, both with or without exercise, but there's a synergistic effect when combined, can protect the brain. For example, one study reports it might help to delay the progression of some major neurodegenerative diseases like Alzheimer's. In elderly patients, creatine supplementation reportedly improved cognition and memory in all but one test, providing additional suggestive evidence that creatine can potentially aid cognitive function in the elderly. Interestingly, the creatine was reported to improve IQ, memory, and working memory in vegetarians. Creatine is stored as phosphocreatine, and this acts as a high-energy reserve. The phosphocreatine decreases rapidly during brain activity, so supplementing with creatine at 2 grams per day for 1 month was reported to increase the average brain creatine level by 9.7%. This finding suggests that creatine can act as an energy source for the brain to use during demanding cognitive tasks like learning, as well as potentially provide an energy source for the storage of newly formed memories into long-term memory. Some of the other potential mechanisms that have been suggested to be behind the effects of creatine include protecting against beta-amyloid toxicity in the hippocampus, and protecting neurons by maintaining mitochondrial energy production, and protecting the mitochondria by raising mitochondrial membrane potential, reducing intra-mitochondrial levels of reactive oxygen species, and reducing calcium levels inside the mitochondria. Glucose is the brain's most important primary source of energy. While glucose has many positive cognitive effects, one review suggests that memory in particular might be especially dependent on glucose for energy. A handful of other studies in both animals and humans have reported glucose can potentially improve memory and attention in college students, improve cognitive function in subjects with different neurodegenerative diseases, enhance learning and memory in healthy young adults and humans, improve verbal memory associated with certain health conditions like Alzheimer's, Parkinson's, and Huntington's disease. Impaired blood glucose regulation has been linked to cognitive impairments, especially visual memory. These potential impairments are reported to be relatively worse in older adults, suggesting age can be an important factor. Because the brain's glucose levels are drained by cognitively demanding tasks, some of these reported cognitive effects might simply be due to restoring the brain's energy supply by resupplying it with the glucose. Alternatively, glucose-sensitive neurons in the brain might serve as sensors of glucose levels. Therefore, the glucose might stimulate these neurons, which may then respond to the signals by stimulating processes related to learning and memory. Some other potential mechanisms that have been suggested to be behind the effects of glucose include improving memory consolidation and memory coding when task demand is high. This is probably just a downstream effect of increased cellular energy levels. Increasing insulin levels, which is linked to memory enhancement. Increasing acetylcholine, which is a critical neurotransmitter for memory and learning processes. Preventing neuronal cell death or apoptosis. Glucose is the brain's main fuel. The brain cells need glucose to carry out cognitively demanding tasks, but the effectiveness and safety of administering glucose to people with cognitive problems is uncertain. Be aware that glucose consumption can contribute to blood sugar dysregulation, leading to diabetes, weight gain, inflammation, and numerous problems. Some decide to go on a ketogenic diet to fuel their brains with ketones instead of glucose. However, this can be a difficult diet, and it might not be the best in the long term. It might be a better idea to alternate between a ketogenic diet and a Mediterranean diet periodically to get the best of both worlds. If you do decide to use the glucose, there's numerous nutrients and herbs that can help with insulin sensitivity and reduce some of the negative effects of the sugar, such as berberine, cinnamon, banana leaf, alpha-lipoic acid, magnesium, chromium, vanadium, fiber, and thiamine or vitamin B1. 24-hour blood sugar monitors or the HbA1c test can help determine your sugar control and your health status. To learn more, you can check out my videos on diabetes. Curcumin has been linked to a number of potential beneficial effects on the brain, as well as on certain health conditions like Alzheimer's disease. In older people, the long-term supplementation with curcumin has been reported to significantly improve working memory. It may protect against stress-induced impairments of brain development, and it might prevent learning and memory deficits. In addition to some of the early evidence suggesting possible effects of curcumin on cognitive processes, curcumin has also shown early promise as a potential preventative supplement for Alzheimer's disease. Some of the early animal findings include curcumin has been reported to reduce spatial memory impairments in rats caused by the buildup of amyloid beta, a neurotoxic byproduct that's believed to be one of the main factors behind the development and the progression of Alzheimer's disease. Curcumin reportedly protected against neuronal cell death or apoptosis in rats, and this might be linked to some reduced impairments in learning and memory. Although the mechanisms behind the potential effects of curcumin have not been conclusively determined yet, some potential mechanisms have been suggested, including reducing levels of TNF-alpha and INOS in the hippocampus. In rats with traumatic brain injuries, curcumin has been reported to help protect and preserve cognitive function by increasing key growth factors like brain-derived neurotrophic factor, CREB, and synapsin 1, by alleviating inflammation by inhibiting pro-inflammatory cytokines, and by reducing oxidative stress. Also by increasing synaptic plasticity, protecting against chronic stress, protecting against glutamate overexcitation or excitotoxicity, and restoring glutathione levels and stimulating insulin receptors. Early human studies suggest that curcumin might prevent cognitive decline and improve memory, but more research is needed. Alpha-GPC is a natural compound found in the brain. It's been investigated for its potential in treating Alzheimer's disease and multiple forms of dementia. According to one study in mice, the alpha-GPC was reported to improve a wide range of memory functions, including working memory, verbal memory learning, and pathway learning in a maze task. Similarly, other animal studies reported the alpha-GPC can improve learning and memory in rats with experimentally induced amnesia. Some of the potential mechanisms that have been suggested to be behind the effects of alpha-GPC include increasing growth hormone levels and increasing acetylcholine levels. Many common lifestyle and dietary factors that can affect how efficiently the brain works and by extension can affect specific cognitive functions like memory. Some of the following strategies have evidence for boosting memory and could be considered as complementary approaches. You can reduce and manage the overall stress, increase antioxidants, omega-3 fatty acids, folate and other B-complex vitamins, medium-chain triglycerides or coconut oil, have regular exercise or physical activity, do advanced cognitive training for independent and vital elderly, known as active, have proper nutrition, do mental exercises and cognitive stimulation, and drink tea, like black oolong tea. To learn about more ways to enhance performance and brain power, you can check out my videos on increasing BDNF, increasing mitochondrial function, increasing circulation to the brain, and my videos on decreasing the risk of Alzheimer's disease and dementia. To book a consultation or to learn about herbs, supplements, or natural treatments, check out my website.